Swayam Prabha. Digital India. Educated India.
Okay. All right. So let us begin. So in the last class, we learned about carbohydrates and proteins. The very basics we learned, and I told you we will learn more elaborately later when we get to those specific chapters. So today, what we are going to do is we are going to continue that, the basic introduction to the other molecules of life. So the next molecule in this is nucleic acids. Okay, so nucleic acids is what we are going to look at. So nucleic acids, again, just like how we learned in carbohydrates and proteins, we'll first begin with the functions of nucleic acids. Because I always believe that once we know the importance of a molecule for life, then we pay more attention to understanding its chemistry. Okay. So this one is a common knowledge. Anyone who knows English, I'm sure already knows nucleic acids are the genetic material. Okay. So they are the a repository of biological information. So what is biological information? So the information required to make a particular biological function. For example, if you take human beings, only human beings give rise to human beings, and at the same time human beings cannot produce other organisms, and other organisms cannot produce anything other than their own kind. So all this tells is every species, for example, Homo sapiens, we have the information in our genetic material to make another Homo sapiens. Okay, and that is what we call as the biological information. When I say make another Homo sapien, meaning all aspects of it, the anatomy, morphology, functions, everything. Okay, so the information to make, or the blueprint to make another organisms of the same kind is stored in nucleic acids, and that is why nucleic acids become another really important molecules. So that's their primary function. So they are not simply storage of information. They have in their structure, there is the ability to copy and transmit that information to a subsequent generation as well. So that is the importance of nucleic acids. That's their main function: storage and transformation of biological information. So now let us look at, since it's a biochemistry class, our focus is knowing the chemical structure and the chemical function of these molecules, and then make biological sense out of them in terms of the biological function. So their structure is, again, they're like all biomolecules. They look really large molecules and look really complex, but when you look into the basic principles, you will realize where they are all simple, very simple molecules, and the complexity comes from the way these simple molecules are arranged repetitively and joined into a polymer. So here, if you look at it, on the right side of the slide, you see a long chain of like hexagonal or pentagonal structures joined by a long line, and that is how a polymer of nucleic acid looks. So in carbohydrates, we called that as a polysaccharide. In proteins, we called polypeptide. So here it is polynucleotide. So the repeating monomer is a nucleotide. So where my cursor, you focus on that, and that is a nucleotide. So the nucleotide has three important components, one shown in this orange color. So this is a nitrogenous base because this ring structure has nitrogen, and therefore we call this as a nitrogenous base. They are of two kinds. The smaller one we call the purine because it's the purine ring structure, or pyrimidine. So you have in a hexagon and a pentagon attached. This is a pyrimidine. So there are two kinds. Purine, the big molecule has the shorter name, purine, and the smaller one has the longer name, pyrimidine. And that is one component of this monomer called mononucleotide. And the other component is a sugar. So we became familiar with glucose and fructose. So glucose is a hexose. So here what is present is a pentose, 5-carbon sugar called ribose. Okay. And this attachment you may not find it hard because we know that the sugar molecule has alcohol groups, hydroxyl groups, and with that you can easily attach the way one sugar is attached to another sugar. And similarly to another hydroxyl group on the ribose, you can have another kind of link. So here it is a phosphoric acid. So it is a phosphodiester. It's an it's an ester linkage, alcohol acid ester linkage. And that phosphoric acid can make another ester linkage with another ribose. And this phosphoric acid, this ribose sugar, and this nitrogenous base, all three put together, as shown in this big enlarged cartoon, forms a nucleotide. Okay, nitrogenous base, pentose sugar, and to that you have a phosphate group attached. So phosphate group has, it is H3PO4. So you have 3 acid groups. Okay, the hydroxyl group here, it is an inorganic acid. This is an acid moiety. It's not to be confused with the alcohol group that you will find in a organic molecule. Okay, so organic acid is carboxylic acid, COOH. So here it is, this H3PO4. These are the 3 acid groups. Of the 3 acid group, here one is in ester linkage with this alcohol group, CH2OH. And the other one is attached to the next one in a long polynucleotide chain. And there is a third one that is still free and that dissociates when dissolved in water, releasing proton. And therefore, this long polymer is actually an acid, because every one of this phosphate group in this long chain has one acid group free, and that dissociates, giving proton in solution. So therefore, this polynucleotide is actually an acid. That is why we call it as nucleic acids. Okay. So now let us get into a little bit more detail. I told you nitrogenous base, 2 types that is visible in this cartoon, like you have pyrimidine and purine. So now let us look at what are the different purines and what are the different pyrimidines. So here are the 3 pyrimidines shown in the top left orange box. So cytosine, thymine. So these 2 are present in the nucleic acid where the information is stored and whose replication helps in transmitting to next generation, and that is DNA. So, okay, so in DNA you have cytosine, thymine. So these are the 2 pyrimidines. In another kind of RNA called ribonucleic acid or RNA, you have, instead of thymine, you have uracil. So in RNA you find cytosine and uracil instead of thymine. And purine, in both the kind of nucleic acids, they are the same 2 kinds. One is adenine and another one is guanine. So here you have the structures. So you see the nitrogen atoms present in these molecules. So that is why this is called nitrogen. These are called nitrogenous bases. Okay. So now I introduce this new term RNA, and then I first mentioned DNA. So what's the difference between the 2? The difference is quite simple. If you focus your attention on this second carbon, so we already learned in carbohydrate class, this is 1, 2, 3, 4, and there it was hexose, and therefore there was one more, and this was 6. So this is pentose, so this is 5. So the second carbon, if it has a hydrogen instead of hydroxyl group, so therefore ending up having two hydrogens, it's deoxyribose, because ribose normally has a hydroxyl group here. And if it doesn't have the hydroxyl group, it is called deoxyribose. And if that is the kind of ribose present in a long chain, then that is deoxyribonucleic acid, and shortly DNA. And instead, if a normal ribose is present with its hydroxyl group in the second carbon, then we call this as ribonucleic acid or RNA. Okay. So one other important information to learn here is, we don't call this carbon numbering as 1, 2, 3, 4, 5. Instead, we call them 1 prime, 2 prime, 3 prime, 4 prime, 5 prime. The reason we number the carbon of sugar in nucleic acid in this manner is because these purine and pyrimidine moiety atoms also need to be numbered. So these are numbered 1, 2, 3, 4, 5, 6. Okay. And as a result, to distinguish between this from this, so the nitrogenous base atoms are numbered, the ring atoms are numbered 1, 2, 3, and the sugar atoms are numbered 1 prime, 2 prime, etc. Okay. So this is the basic structure of a nucleic acid. Quite simple. All we need to know is it is a polymer, and the repeating monomer is a nucleotide, and the nucleotide itself has three components, namely a nitrogenous base, a pentose sugar, and a phosphate group. And this phosphoric acid makes two ester bonds, and therefore we call as diester bond, between adjacent ribose sugars in a long chain. And this is what we call as the sugar phosphate backbone. Okay, so the polynucleotide is held together primarily by the sugar and the phosphate. Sugar, phosphate, like this. And these are attached to the sugar moiety, the nitrogenous bases. Okay, so this is the basic chemistry of nucleic acids. But their structure, from the structure, an emergent property arises and emerges, and that is shown in this slide. So if you have one polynucleotide chain and if you have another one adjacent, the atoms of the nitrogenous bases, so the nitrogenous bases project towards each other, and the atoms of the nitrogenous bases come close enough for hydrogen bonding to happen. Okay, see here you have this, strongly electronegative oxygen. If a hydrogen comes from nearby, then you can have a hydrogen bond here, and so on. So if a guanine and cytosine come adjacent to each other, there are three hydrogen bond possibilities. So in this introductory class, I'm not getting into which are the three atoms here that contribute to the hydrogen bonding. And similarly, if an adenine comes adjacent in the other strand, adjacent to a thymine, then two hydrogen bonds are possible. And these hydrogen bonds individually, although they are weak, but together in a long chain, they are of significant force to hold two polynucleotide chains together as double-stranded structures. Okay, so the DNA present in our genetic material, like for example in our our cell nucleus, they are present in a double-stranded form. One polynucleotide adjacent to another one, and the bases stack up in the middle, in the center of the two strands, held together by hydrogen bonds. And this sort of a structure, that the nitrogenous bases do not project outward, like, this side and this side would be outside. Instead, these are facing each other, and this portion is called inner side, and facing inside and allowing these the hydrogen bonds to form was proposed by Watson and Crick, and therefore these hydrogen bonds are often called as Watson and Crick base pairs or Watson and Crick bonds. Okay, so this is what makes them to be in double-stranded form. And in this stacking arrangement, they don't remain like a long ribbon. Instead, they form a helical shape shown here. Okay, so they form a helical structure with one turn, like from here to here. They hold about 10 base pairs. Okay. So adjacent base pairs are of 0.34 nanometer distance, and therefore one turn of this helix is about 3.4 nanometers. So this is called the major groove, this is called the minor groove. So those details today I'll ignore. When we get into the nucleic acid structure, probably we won't be doing that in the biochemistry class, but when you learn molecular biology, where you will learn more about DNA replication, transcription, etc. There you will learn about these structures as well. But for this class, all you need to know is polynucleotides contain nucleotides as the monomer, and the nucleotides have 3 components, and 2 adjacent strands can base pair, and that base pairing is these are bases and they pair, like A, T and GC, and that's why it's called pairing, and that base pairing bond is actually hydrogen bond. And this sort of a double-stranded structure forms a helical twist, and that is how the actual DNA structure is. So this is what we need to know. So one important additional information we should know is there is a directionality for these strands. Okay. So let's go a little bit back and come to this again. Yeah. So here, if you look at it, so if you take this ribose, let us say this is at one end of a long chain. So that means here it is no longer, it is not attached to any more nucleotide towards the bottom of this slide. So that means this hydroxyl group, this 3' hydroxyl group, so anyway, why it's not going away. So this hydroxyl group here would be free. This is not esterified with any other phosphate. So this will have a free hydroxyl group at this end. This 3' carbon will be free hydroxyl group. On the other hand, at the other end of this chain, so you have here, therefore this is the 5, this corner here is this 5' carbon. So 5' has a ester linkage. This one has 3'. So it's 5', 3', 5', 3', like that it goes. At the very end of this ribose at its 5' corresponding to this carbon has only one phosphate attached, and it is not linked any further, or it may not even have a phosphate in some situations. Okay, and this end where the 5' is free, we call the 5' end of the chain, and this end where the 3' hydroxyl is free, we call 3' end of this chain. So this particular ribbon shown here runs 5' to 3' in this direction. So if another molecule comes here exactly in the opposite orientation, suppose if I reverse this and have 5' free here and 3' free here, it would be anti-parallel to this. And that is the kind of chain with which this base pairing happens. So one chain or one strand runs anti-parallel to the other one. If this is shown here 5 prime to 3 prime, see this is 5 prime to 3 prime. 5 prime is free, not linked into another nucleotide. So therefore this is 5 prime end. This is not linked to another one, therefore this is 3 prime end. So this 5 prime 3 prime strand base pairs with another one that is 3 prime to, like for the same direction. This is 3 prime 5 prime. This is 5 prime 3 prime in this direction. Okay. So one strand essentially base pairs with another strand that is anti-parallel. Okay. So this is another thing you need to know. So when people talk about a 5 prime end, 3 prime end, you need to correctly understand. Do not think that, in the top of the slide is the 5 prime end and the bottom of the slide is the 3 prime end. That's not how it is. Okay. So molecules are in solution. They have 3-dimensional arrangement and they can be in any orientation. So when we say this, these are all with reference to other constituents of the molecules, not the way we are schematically writing. So that is all we are going to learn about RNA and DNA for this very introductory class. And now we go and familiarize ourselves with the fourth large biomolecule. These are the only 4 we need to learn. So biochemistry is actually not that complex considering the complexity of organisms. So the last class we are going to learn are the lipids. Okay. So again, let us focus on the functions. So lipids are extremely important molecules. Okay, without lipids, we won't be able to exist as we exist now. So they are very important. The most obvious one, easy to understand, is that all our cell membranes, the biomembranes, are made up of lipids. Okay. And lipids present just below our skin, subcutaneous fat, insulates our body internal organs from variations in the temperature in the outside environment. Okay, so they help in insulation. And these are actually the most compact storage of energy. So carbohydrates, we learned, also store energy, right? So we learned that the carbohydrates, like for example, starch and glycogen in our animal liver, I told they store energy. So in addition to carbohydrates, lipids also store energy. Lipids are actually, they store more energy per mole compared to carbohydrate. That is because these are highly reduced, and as a result their energy content is more than that of carbohydrates. So they nearly store double the amount of energy per unit mass compared to carbohydrate. So they are serious storage of energy. For example, if you take all the seeds, like oil seeds, like groundnut, mustard, and so on, so they have stored the energy in the most compact form. Therefore, the seed size need not be very big, and these lipids are then used to derive energy for the seed to germinate and grow. So since this is a very compact form of storage of energy, the seeds use them primarily for storing a large quantity of energy required for seed germination until photosynthesis can be started in the form of oil. So these are really important. So therefore we cannot ignore lipids. So what are lipids? Like, are they again polymer made up of monomers? No. So they are different. So they are large molecules, but they are not polymers. So that's one big difference between the other 3 groups and lipids. So lipids are actually different types of molecules. Some are fatty acids. These are like acetic acid, acetic acid CH3COOH, right? So you have one carboxyl group. Now, if you keep increasing the chain, instead of CH3COH, you have CH3CH2COH or CH3CH2CH2. Like that, if you keep increasing the length of the chain, what you get are fatty acids. So then we will look at the structure in the next slide. And these fatty acids, when you esterified with an alcohol, a sugar alcohol, you get what are called triacyl glycerols. And to them, if you add a phosphoric acid, you get phospholipids. Then there are some aromatic rings called sterols and then waxes. So these are the 5 major kinds of lipids we have. So lipids are also known as fats. Okay, so FAT, fat. So they are synonymous. So we will look at each one of these, how they look like and how their unique structures help the current kind of functions they perform. Okay, so these are fatty acids. So you have a carboxylic acid, so that's why they are acids, and a long chain of aliphatic group. Okay, with the carbon's valency, all you need to do is just to fill it by adding hydrogens. Okay, the last one is CH3. So when you have a chain like this is called saturated fatty acid. Okay, and if you have a double bond in the chain, and this is a degree of unsaturation, like one bond is unsaturated, so we call this as monounsaturated fatty acid, and polyunsaturated when you have multiples of them. Okay, and these molecules are essential. The polyunsaturated, monounsaturated are very important, and and some of them cannot be made in our body. So they are usually made by microorganisms and plants, and therefore they are to be present in our food, and we call them as essential fatty acids. So don't worry about that for now. So all you need to know is fatty acids can either be saturated or they may be unsaturated, and depending on the degree of unsaturation, we call them monounsaturated or polyunsaturated. So these are often called free fatty acids because these carboxylic groups are not linked to any other molecule. Okay, so as a result, they are called FFA. So if you take any food, like if you take a biscuit packet and look at the ingredients, and some of them where the labeling is very professionally done, they will tell you FFA, how many grams per 100 gram of that biscuit. So FFA stands for free fatty acids, and under that they would have labeled MFA or PUFA, PUFA, polyunsaturated fatty acid. They would have written that. So you can look at any food packet, any packaged food, if you take and look at the composition or ingredients, nutritional facts, and there you will see that. And so I told you they are free fatty acids, and then the carboxylic acid may be linked to other molecules, and one form of linking leads to what we call triacyl glycerol. This is the form in which our cells store fat. Okay, so fat is stored sometimes to some limited quantity in liver as well. Some of the diabetic patients have large storage of fats in the liver called fatty liver, and most of the normal people, we have fat stored in adipose tissue. That's a sort of tissue that is present lining the internal organs in the body cavity and primarily under the skin, subcutaneous fat. And that is primarily in this form. So here the fatty acid, the acid group here is the hydroxyl group. Okay. That is because it is not merely hydroxyl group, it is attached to a carbonyl carbon here. So as a result, it has acid property, not alcohol property. And here you see this hydroxyl group, another organic molecule, this is alcohol. So this is an ester linkage. So glycerol is actually a sugar alcohol. Okay, so it looks more like the glucose. Instead of 6 carbon, it is having 3 carbon. The difference is it doesn't have that aldehyde group here. Okay, and all of them have an hydroxyl group attached. So we call this as a sugar alcohol. Instead of aldehyde, here you have an alcohol group. Aldehyde or ketose group, you have an alcohol group. So therefore it is a sugar alcohol. So with these 3 alcohol groups of glycerol, when you have 3 fatty acids linked, you call that as triacyl glycerol. This carboxylic acid moiety is called an acyl group. And you have 3 acyl groups attached to glycerol. So this is glycerol derivatized with 3 acyl groups. And that is why it is called triacyl glycerol. Okay. So butter, for example, shown in this plate is primarily triacyl glycerol. Okay. So from triacyl glycerol going to phospholipid is no big deal. It's a small change in the structure. And here I have shown its importance in terms of the function. I always get fascinated by the beauty of this molecule. So what it does is, suppose if you look at this structure. So here, the bulk of the molecule is essentially this aliphatic group. There are no polar groups here. It is totally hydrophobic molecule. Okay. So it is just not going to dissolve in water at all. So as a result, it is very useful to compartmentalize aqueous environment. Okay, aqueous meaning water based. So our cytoplasm, like the inner part of the cell, is aqueous. So separating one cell from another cell means you need something that can partition or compartmentalize water-based solution. So that means the compartment has to be something that is hydrophobic, and that is served well by this hydrophobic nature of these fatty acid tails of triacyl glycerol. But that is okay in normal, artificial human being generated world. But biology is a lot more sophisticated. So here the lipid does not serve merely as a partition. It actually can interact upon need. So that is provided by derivatizing this molecule. So instead of this group having a fatty acid attached, this alcohol group of glycerol, instead of esterifying with another fatty acid, it is in an ester linkage with an inorganic acid, which is the same phosphoric acid that we saw in nucleic acids. So instead of this third fatty acid, you have a phosphate attached. And once you have phosphate attached, it is called phospholipid. That's the difference between triacyl glycerol and phospholipid. So phospholipids have 2 acyl groups in glycerol, and the third one is a phosphoric acid. And the other 2 acid groups of phosphate may be now derivatized with attaching to other polar molecules. And they are the very basic phospholipid has just the phosphoric acid, and that is called a phosphatidic acid. We will learn that in detail when we go to the lipids class. So for now, all you need to worry is in triacyl glycerol, the last free fatty acid is replaced with a phosphoric acid to make a So this molecule becomes amphibethic. It has a hydrophobic group as well as a hydrophilic group. And that is what you are seeing here. So if you look closely on these round structures, each one of these yellow round structures, you will see two tails jutting out, and each of those two tail-like structures are nothing but these two groups. And that yellow itself is this glycerol with its phosphoric acid and other hydrophilic things attached to that phosphate group. And that is what we call as the head group. So we call that as head group, and the two fatty acids as the tail group. Okay. And these tail groups point towards another layer of phospholipid molecules, and these hydrophobic things interact among themselves through hydrophobic interactions. And these head groups being hydrophilic, they can interact with the water-based aqueous environment. So in this manner, phospholipids help in compartmentalizing aqueous environments, but at the same time, the compartmentalization comes from this, okay, the two acyl groups here. At the same time, they are not in, they are in talking terms with these aqueous environments. They can interact through this. So basically, it's a wettable surface. This head group can interact with water, and this head group can interact with water this side. And the two layered membrane or bilayer is what is our cell membrane is. So this is the importance of the phospholipids. Do not worry about these structures. We will learn about them later. These are basically proteins embedded in the lipid bilayer. So our cell membrane is nothing but these phospholipids. So each phospholipid is essentially triacylglycerol, in which the third alcohol group of glycerol is esterified with a phosphoric acid, and that phosphoric acid may have other polar groups attached to it. And that is how phospholipids themselves are many kinds of them. So that is phospholipid. And the last class of not the last one, the last but one, are the sterols. Okay, so what are sterols? Sterols, the very basic kind of sterols present in our in living organisms is cholesterol, and all sterols, including cholesterol, have this ring structure. So this is called cyclopentane, and this three aromatic rings fused in this fashion is called a phenanthrene. So this basic ring unit is called cyclopentano-phenanthrene ring. Okay, so this is basic for all the sterols in our body, and one sterol varies from another sterol in the other constituents, like this side chain and presence of these methyl groups, this double bond, this hydroxyl group. When you have all of this, it is cholesterol. If you have some other group, then it is some other name. Okay. Sterols are also very important. Many signaling molecules in our body, including the two hormones that are responsible for being male or female, is sterol molecules, testosterone and estradiol. So they are derivatives of cholesterol. Okay. So that is all we are going to learn about sterols right now. So sterols are made up of this four rings formed in this manner, and they may have derivatives, and the basic one is cholesterol. And cholesterol again is a storage lipid and extremely essential important molecule. So you would have learned about cholesterol already in the common newspaper, and your friends and family talking about cholesterol being bad, and somebody's cholesterol is high, and as a result that person has diabetes or hypertension, etc. But those issues are only when the normal molecules have gone in the wrong way, when they are too much or their production and degradation is not managed right, that's when you have abnormality. Otherwise, these molecules are absolutely essential. Cholesterol is embedded in these this phospholipid bilayer, and that's very important for our membrane function. And in addition, this is precursor for all the steroid derivative hormones, and those hormones are important signaling molecules. Okay, so therefore they have very important functions. The last group of molecules we are going to learn are the waxes. And waxes are extremely hydrophobic. Uh, they are simply long chain of fatty acids, which we are already familiar. And assume another long aliphatic chain at the end. Instead of having carboxylic acid, suppose it as an alcohol group. Okay, they are called fatty alcohols. So when you have such a long fatty alcohol and a long chain of fatty acid linked in ester bond, you call them waxes. And the waxes coat the surface of the leaves, like some of the shiny leaves have that, and due to that they are extremely hydrophobic and protect the leaves from transpiration. They don't readily lose moisture due to evaporation, so they give that protection. So that is why the leaf surface is shiny. The shininess comes from the coating of wax. So for us as biochemists, our the information about wax is they are esters made up of long chain fatty acids with long chain fatty alcohols. Okay, they are also they are stored in marine animals like whales, and they are important source for making fragrances also. Okay, so that's about the waxes. So we are sort of running out of time, so I'll stop here. So we have learned today the basic biochemistry of nucleic acids and lipids, and we looked at the different kinds of lipids. So in the next class, we'll move on to the next topic, where our goal is to understand how the arrangement or the way in which the constituents of a molecule are arranged in space, in the three-dimensional space, become very important in biology. Okay, in chemistry it doesn't matter, the reactions happen through the functional group. So as long as a given functional group is there, the reaction is going to happen. But in biology, simply the presence of functional group is not enough. The actual arrangement of atoms in a molecule, the three-dimensional arrangement, becomes important, and that is what we call as stereochemistry. So we will learn that topic in the next class.